Okay, hello everyone, and welcome to our very first podcast episode. Today, I will talk to our very first guest, who is Christina Dickmeis. And Christina works with Pichia pastoris to express protein. And she will tell us a lot about her project today. And yeah, I think it's best if I let her introduce herself. So hi, Christina, and thank you for being with us today. Can you tell us a bit about yourself and what you are working on right now?
Yes, of course. So I'm Christina, and I'm working as a senior research scientist here at SBI. And I'm working in the R&D department and try to develop our new sensors that we want to add to our portfolio in the future. So my main tasks are to produce enzymes that we can use for the development of the new sensors that we want. And I'm modifying these enzymes to our needs, and then I try to find the optimal expression host, test different modifications, test the production in the host that we then decide on, and yeah, of course, also develop the production strategy and the purification strategy for the later applications, and also test the immobilization to different sensors that we want to use.
Mm-hmm. That sounds very interesting. And if you think about the stage of your project right now, could you tell us what the aim of your current project is at this stage?
Yeah, so we decided for the first enzymes that we want to use. So I'm currently testing the optimal expression host. We decided now to go for Pichia pastoris. So I'm trying to find an optimal production strategy and to increase the product yields for an efficient production process.
Mm-hmm. So you want to increase the yield of your protein to have more target protein in the end. And what would you say is the main challenge of this?
Yeah, so currently I'm doing a lot of screening tests in shake flasks. So the main challenge at the moment is more an applicative challenge. So I'm using our liquid injection system and also our cell growth quantifier to find an optimal feeding strategy and to test the most important parameters for my expression clones.
Mm-hmm. And how do you do that? Like how do you induce basically?
Yeah, so we choose to use Pichia pastoris as the main expression host, and we use the classical AOX1 promoter for induction. So we are using the volatile component methanol as an induction medium. So the main challenge is to handle, of course, the methanol, which is toxic in principle, but not for the cells in small amounts. And I'm using this with our liquid injection system, which makes the handling much easier because I only have to handle it once, and then I have all in the setup.
Mm-hmm. So you mentioned that using methanol basically bears some difficulties and that you chose to use an automated feeding system. Can you maybe shortly explain what automated feeding is?
Yeah, so I'm using the liquid injection system. So I add the induction medium that I want to use in a cartridge, which is mounted on top of our shake flask. And then I can use our DOT software to define the feeding strategy I want to use. So I can say, I want a single shot of my induction medium. I can say we want to use multiple shots in certain time steps. I can say I want to use a constant feed or an exponential feed. And I also can combine these methods.
Okay, and you mentioned that you tested basically different expression hosts, right? Can you maybe tell us which systems you tested and what the differences between these systems were and which advantages and disadvantages you saw?
Yeah, so we started using E. coli as a rather simple expression system because it's very well described, and in theory should also work with the chosen enzymes. But there I had the problem that although the enzyme yields were very high, so the expression worked really well, but I had inclusion bodies in E. coli. So I needed to purify inclusion bodies and then refold the enzyme with the suitable cofactor. And that took quite a long time, and the refolding success was very low. So I lost a lot of the enzyme, and overall this process was not really economical, feasible for our needs. So we switched to Pichia pastoris as a eukaryotic system, which also has all the post-translation modifications that we need, for example, glycosylation, like it's in the natural host, although it's of course not exactly the same glycosylation, but the glycosylation improves the stability of the enzyme. I have a correct folding of my protein in this host, and also we can use the secretion system in Pichia, so the protein is directly secreted into the medium. That makes the purification much more easier because we have much less host cell proteins that we need to get rid of during the purification steps.
Mm-hmm. Yeah, so that sounds understandable that you chose to use Pichia then instead of E. coli. And having decided that, what was the next step in your project?
Yeah, so once we cloned and did all the first steps in Pichia and analyzed that in principle all worked, I'm now trying to improve the product yields. So I'm doing induction screening, so testing different induction methods, different feeding protocols with the methanol that I use. So usually in Pichia, you start with a normal batch cultivation on a medium which contains glycerol or glucose. We choose glycerol for the first carbon source, and once the growth is finished, you switch to methanol as carbon source and also as an induction medium. So I'm now testing different feeding strategies. So when you work in shake flasks, normally the manual handling is done that you change the medium to a methanol medium and then do an induction shot every 24 hours with methanol. So you have every 24 hours, 1% methanol in your culture, which is used, and then you just give new methanol. And each time you have to stop your process, transfer the shake flask to the clean bench, and open the shake flask, give the methanol. So you have to disrupt the process, increase the risk of contamination. And I'm now trying to do all these tests or all these screenings with our liquid injection system, so I don't have to stop the process, and I can test different feeding strategies at once from the exact same starting culture.
So these different feeding profiles that you were talking about, can you tell us what feeding profiles they were? How did they differ between each other, and why did you choose these feeding profiles to test?
So I choose the setup for my experiment that I start with a pre-culture. I inoculate a large amount of culture volume and then divide this culture volume into the different shake flasks. Then I mount the list cartridge with the methanol amount that I wanted to use to the shake flask and transfer all these ready set up shake flask into the shaker, and then start the experiment. And I then also use our cell growth quantifier to monitor the cell growth. And I choose to start the methanol feed independence of the biomass. So I used a biomass-based feeding. So once the cell growth stops, all the glycerol is used up, and the growth rate goes near zero, the methanol feed is started. And then I compared the normal strategy that you would use. So use one shot every 24 hours, like you would do it with a manual handling. Then I tested giving one shot every 12 hours, but with an overall smaller volume. Then I tested giving one shot, then wait for 12 hours and then start a constant feed. And I also tried to use a constant feed from the beginning. Overall, I also kept the total amount of methanol that I used constant in all my experiments, so that I really would only have as a variable the different feeding strategies that I tested.
Mm-hmm. You mentioned the method of biomass-based feeding. Can you explain what these terms mean in general?
Yeah, so with the combination of the cell growth quantifier with the liquid injection system, I can define or say via the DOT software when my feeding should start, independence of my biomass. So I can either use a certain threshold of my biomass to start the feedings, or I actually prefer using the growth rate, because that's a quite accurate signal. So I can say, once my cell growth is either above a certain threshold or beyond a certain threshold, to start whatever feeding I want to start. And I can also say, okay, I know at the beginning of my growth, I have a lot of little variations, so only start this after a certain time point. So for my Pichia experiments, of course, I know the first growth will take at least 24 hours or longer on my starting medium. So I say, okay, start with my feed only after 30 hours if the threshold is reached. And I can also say, just wait for a maximum time. So if under which circumstances ever this threshold is not reached, start the feed anyway. So after 48 hours or so, definitely start the feed.
Mm-hmm. Okay. So basically you have like security trigger points, let's say, to make sure that the feeding really starts. Yeah. Okay. It seems to be quite some work until you finally move to the bioreactor. So in your opinion, why do you think it is important to do all these optimizing steps in shake flask before you can finally move to the bioreactor?
Yeah, so especially for smaller labs, I think it's very time consuming and quite a lot of investment to do all the screenings in the large scale bioreactors. So if you don't have a lot of small bioreactors, you really can't do all this screening in parallel. So I think the shake flask is still the one vessel most labs have available. And when I can do all these screenings in parallel with a very automated system, so I have as less manual handling as possible, so I don't have any interruption of my process. I can monitor the cell growth and a lot of parameters. And also, I don't have to interrupt the process and have a higher risk of contamination. Can really ease all the developments before actually transferring into the into the bioreactors, which is much more handling time. So when I start a process in the bioreactor, I need at least 1.5 weeks for preparation, doing the actual process. In Pichia, the induction time is usually chosen between 72 and 96 hours, so quite a lot of time. And also the cleaning afterwards, the sterilization process takes quite a lot of time. And so it would take me several weeks if I only have one bioreactor available to do all this screenings, like find an optimal adaptation time that I can do in parallel now in my shake flasks.
You already mentioned a bit why it is helpful to have less manual handling, basically. But can you maybe elaborate a bit why you think automation in shake flasks is so important?
Yeah, so at the moment, most screening processes in most of the labs is performed in shake flasks, but the optimization for scale up is still very complicated, and mostly because of the very different oxygen transfer rate between the shake flask and the bioreactor, the actual scalability is rather low. And if we gain more insights and have more automated systems, we hopefully can close the gap for the scale up between a shake flask and the bioreactors. A lot of screenings, of course, is also performed in microtiter plates, but therefore a lab also has to do a lot of investments to have all the system available for this microtiter plates, and doing all this assays and automation in these types of vessels. And the shake flask is for most labs still the starting method of choice. And we hope with our systems in the future we can close the gap for the scalability from the shake flask to get into the bioreactor.
Thank you very much for all your insights today. And I hope to see you and speak to you again soon.
Yeah, I hope as well.